Okay, guys, so we're going to talk a little bit about genetics and heredity. In this set of videos, again, this is kind of going to be more based off of your bio, like 100. A lot of the stuff is going to be based off of that. And we talk about it because we were talking about reproduction, and so therefore you kind of have to understand like what is the human genome and how does that play a role in determining who you are. Okay. So essentially, whenever we talk about any type of genetics, we're going to talk about the scientific study of how things are inherited. So explaining how certain characteristics are inherited from one person from mother to daughter, or from father to daughter, or whatever the case may be. We're mostly when it comes to genetics and what kind of in general what um, motivates people to study genetics in the first place is going to be how those diseases are directly inherited, because you do have diseases that are able to be inherited. Okay, so whenever we look at genes, we want to know what the genetic code is. So this is kind of like what your DNA that codes for a specific gene that's on your chromosome. Okay, so that's the first step. The second step is going to be what your chromosome is. So chromosome, that's going to be whatever that's going to have your DNA. They really only exist during cell division because they've been replicated until they're very thick. And they kind of, form that little chromosomal, thing that only occurs during cell division. So this is kind of when we're able to best get a look at what our chromosomes are supposed to look like. And then chromatin is going to be a DNA, strand of form of DNA that's made up of subunits called nucleosomes. Again, that's basically how we move our genes from one cell to the next. So each gene is going to be a sequence of nucleotide bases in a DNA molecule, and those basically transcribe for your RNA molecule. Every RNA molecule codes for a protein. Okay, so which occurs in your ribosomes. So you have DNA that codes for the RNA, which codes for the protein. So essentially, DNA is your gene, protein is the expression of your gene. Okay, so that's going to basically how you determine what the structure and function is. Basically, is through your structural proteins that are found within your DNA. So the human genome. So it wasn't until recently that we really started using DNA in general, and the entire human genome wasn't really completed until 2003. So in general, we contain about 19,000 genes. Okay, so when we look at our different chromosomes that we see, whether we have chromosomes, 23 chromosome pairs, so our 46 chromosomes in general, there are 19,000 genes and non-coding DNA on those 46 chromosomes. Okay? So the idea here is that these genes will encode for different functional products. So whether it's transfer RNA, which is just basically what's attached to the amino acids that build your proteins, or ribosomal RNA, which encodes essentially how the ribosomes are, made during cell division, and riboenzymes, all of that. Any of those, they can also encode for different functions within the cell. So we also have pseudogenes, which are basically bits that we kind of called non-coding junk DNA, but they used to be formally functional genes. So example would be like, if you look at comparing chromosome 1 to the Y chromosome, there's a lot of difference in there, and some of it's non-coding junk DNA. Okay. So this is just basically our different types of the human genome. And again, they're talking about here, whenever you see the different, colors here, that's determining the karyotype. It determines where exactly those genes occur. Okay. So each one of those might be a specific gene for a specific trait. Okay. As best that they can tell you. Okay. So chromosomes and genes. So basically, whenever you're analyzing the genome code to determine what makes someone different than another, and to determine where exactly a hereditary disease may occur, you would be doing genomics. And if you're looking at the mRNA codes, you're going to be looking at transcription for the most part. And so therefore, you're going to be looking at the transcript. It's called transcriptomics. And then if you're looking at the proteins, you're going to be looking at proteins. And that's going to be a different thing as well. Okay? So genetic information, genomic information can be expressed in a variety of different ways. So you can do it in like an ideogram, which basically shows the area along the chromosome that is shows where exactly it is, or they can also be represented as actual nucleotide bases. Okay. So remember we talked a little bit about the last one, you know, PowerPoint or last set of slides. We talked about meiosis and how that occurs. Okay, so we talked about how you go through meiosis 1 and then through meiosis 2, and at the end of it, you end up with only 23 chromosomes instead of the full 46. So the idea here is that this occurs because of the principle of segregation, which allows for the chromosome pairs to separate during the first meiosis 1 or the first part of meiosis. Okay, when you have a sperm and a egg or an ovum unite at conception, they form the zygote because both of those nucleus merge to form the 46 chromosomes. Okay, so again, there's a couple of different things that allow for different traits to occur. You have crossing over and gene linkage. So crossing over occurs when there's genes from one location that cross over on the matching chromosome. And essentially kind of, you have like a little bit of genes from one chromosome and a little bit of genes from the other chromosome. You want to look for exactly how that works. You can see that on page, well, actually, we've got it right here for you. Okay, so this is crossing over. So essentially what happens is that they cross over and we switch and we have a portion of the blue on the red side and the red on the blue side. That's all crossing over is. Now, we can also have gene linkage where genes on individual chromosomes, they may stay together because they're really close to each other. So that's the idea there. So you're going to have some genes that'll be linked together. And that's because they're literally like, if you looked at it on here, it would be like this one gene would be coded here and another gene would be coded here. And so because of how close they are, they're going to stay together. Okay. So this all comes from the idea of independent assortment. And this is that basically it doesn't matter exactly how they're going to separate, but they're always going to separate. And essentially what happens is the maternal and paternal chromosomes will get mixed up and redistributed independently. Okay, so the idea here is that they're not going to necessarily line up all blue on one side and all red on the other side like they show in the picture. You can have red and blue, it doesn't really matter. They're not going, no one chromosome is going to be attached to another chromosome. The only thing that'll be attached at all will be the gene linking between two individual chromosomes, where genes that are so close together will stay together. That's the only thing that'll be linked. And then genetic variation. This is what all of these things occur to. They allow for genetic variation because it means there's not going to be a specific offspring that's going to look exactly like their parents. There's going to be something that's going to be different about each one of them because there's, you have your genes, and then you have the father's genes as well. Let's say. And that would be, you're going to be looking at also seeing your parents' genes mixed up in there because you have different chromosomes from your parents that have also been crossed over. So therefore, you could be, you could have almost, genes that are linked all the way back to your great-great-grandparents. But they could be from one side and the other side all on the same chromosome. So, okay. So when it comes to looking at genetics in general, you have dominant and recessive genes. So dominant genes are going to be kind of like, these are going to be your genotypes, and they tell you essentially what your phenotype is going to be. So your phenotype is what you actually express. Your genotype is what the, code or the dominant and recessive genes. Okay. So dominant genes, no matter what, they're always going to see those effects. Recessive genes, you're only going to see them if they're both recessive. They're masked by the dominant gene. Okay. So you're going to get one from each parent. So this is an example here. We've got albinism. So if you look at it here, we've got a mother carrier and a father carrier. So what happens is that essentially you have one that has the dominant regular normal pigmentation and the coding for albinism. And essentially you also have the same thing here with your father. And for one offspring, you could have normal pigmentation because they're both dominant genes, so therefore the dominant gene will be work. You can have another carrier who will have normal pigmentation because, again, the recessive gene is being masked by the dominant gene. And then the albinism is when the two recessive genes come together. Okay, so this is determined by your genotype. Okay, this is going to be the combination of whether or not you are, recessive or dominant. And this will occur whether you have homozygous or heterozygous. So homozygous means that you have two of identical traits, essentially two of the same. So you either have two dominant genes or two recessive genes. So this would be the example of the normal pigmentation who was not a carrier, and the albinism. Those would both be considered homozygous dominant and homozygous recessive. And then heterozygous, that's a genotype with two different forms of a trait. So that means the carrier here, the uppercase A and lowercase a, that is heterozygous. And remember, again, homo means the same, hetero means different. Okay, phenotype, that's just going to be how it's expressed. So essentially what your proteins are going to express. So whether or not you have normal pigmentation or albinism. And then you can also be a carrier. So that means that someone who has a recessive, and this is only occurs for recessive gene diseases. So any type of recessive traits that are code for specific inheritable diseases. That would be a situation of, or heritable traits. That would be a situation where you can have a carrier, which means that you have for the dominant gene, it covers up the other issue, but you could possibly have an issue if the recessive gene and you're with another carrier, the two recessive genes come together. Okay, so when we're looking at polygenic traits, essentially what those are is that we can't specifically link them to one specific gene. There's a number of different genes that code for the specific trait, and that can be a simple situation of hair color. There's too many things because there's so many different things that determine your hair color. So therefore, there's several different genes that determine your specific hair color. Co-dominant, that means that essentially when you have two different dominant traits that occur together, each will have an equal effect. Okay, so that would be kind of, if you think about it, AB blood type. That is an example of two that are co-dominant because A is also dominant and B is also dominant. So you express both, which means you have the antigens for both of them. Okay, on your blood cells. So sometimes the abnormal disease genes will actually persist in a population to provide some type of biological advantage, and it's usually kind of like a survival of the fittest situation. So again, sickle cell, the reason why that occurs because malaria, was rooted in Africa as its point of discovery. And Africa is known to have African, darker colored skins, and the African population is known to have sickle cell anemia. And so therefore the sickle cell gene actually protected them against malaria. Okay, so as you can see there, the frequency, and it's all based off of the distribution of malaria. Okay. So sex-linked chromosomes. So sex-linked traits. So remember, your female chromosome is going to be the X chromosome. The Y chromosome is the male chromosome. So females can only give you the female chromosome. That's going to be the X-linked chromosome. It's much larger than the Y. So usually anything that's on the female chromosome when it comes to genetic diseases will be portrayed if they are X-linked if you have a Y chromosome with it. If you have two X-links, you're not going to see them because the regular X chromosome will probably most likely overpower the other X chromosome. Okay. So this is why we call sex-linked traits. Again, it's things that are on the sex chromosome. So here, if you remember, the Y chromosome is much smaller than the X chromosome, and so therefore the X chromosome almost overpowers them. Okay. So when we look at this here, here we can have accentuation color blindness as an example of a sex-linked trait. It's recessive, so therefore the regular X chromosome is going to overpower the other X chromosome. So the mother here is going to be a carrier. This means that this X here has the extra abnormal X chromosome, and therefore if this X goes with this X from the male, from the father, she's going to have another offspring that's a carrier. But if this X goes with a Y chromosome, then that means that the male is going to be colorblind. And then obviously, if we have regulars, we're going to have a normal male and a normal female. Okay? So I'm going to go ahead and pause it right here. And then we're going to talk about some genetic disease. I haven't really been super strict about that for this semester.